Good evening, and welcome to Cozy Sleep Time. Before we begin tonight's story, take a quiet moment to settle in and get comfortable. Find a position where your body can fully relax, whether you're lying in bed, resting on a couch, or simply unwinding after a long day. Allow your shoulders to soften. Let your breathing slow down just a little. There is nowhere else you need to be, and nothing else you need to do. Here on Cozy Sleep Time, we explore calm and gentle topics. Stories designed to help the mind slow down and drift peacefully toward rest. Each story moves slowly, gently, allowing your thoughts to wander and your body to relax more and more. Our story tonight is about the hidden life of soil. As the story unfolds, you can simply listen, or you can allow your mind to drift. There is no need to stay focused. If your thoughts wander, that is perfectly natural. So take one more slow breath, get comfortable, and let the quiet story begin. Beneath our feet lies a world more alive than any forest above. A single teaspoon of healthy soil contains more living organisms than there are people on Earth. This hidden universe of microscopic life performs one of the most essential functions on our planet: the decomposition of organic matter and the recycling of nutrients that every living thing depends upon. Soil is not merely dirt, it is a living, breathing ecosystem. Among its countless inhabitants are bacteria, fungi, protozoa, nematodes, mites, springtails, earthworms, and thousands of other species that rarely cross our awareness. Each plays a distinct role in breaking down dead material and transforming it into forms that new plants can use to grow. Bacteria are the most abundant life forms in soil. A gram of fertile soil may contain hundreds of millions of these single-celled organisms. They specialize in breaking down simple sugars and carbohydrates, the easy-to-digest components of dead plant and animal matter. Without bacteria, leaves would not decompose, and the forest floor would accumulate with dead material that new growth could not penetrate. Fungi complement bacterial activity in essential ways. While bacteria handle the easily decomposed sugars, fungi tackle the tougher materials. The cellulose in plant cell walls, the chitin in insect exoskeletons, and the lignin in woody tissue all require fungal enzymes to break down. Some fungi extend their hyphae through soil like a vast network, transporting nutrients and water between different parts of the ecosystem. The relationship between fungi and plant roots is one of nature's most important partnerships. Mycorrhizal associations form between roughly 90% of all plant species and underground fungi. The plant provides the fungus with carbohydrates from photosynthesis, while the fungus gathers phosphorus, nitrogen, and water from the soil and delivers them to the plant's roots. This exchange sustains both organisms and shapes entire communities of plant life. Protozoa are single-celled animals that feed on bacteria. They play a crucial role in nutrient cycling by releasing nitrogen and other nutrients from bacterial cells when they consume them. The released nutrients then become available for plant uptake, completing a cycle that moves essential elements from decomposing organic matter through bacteria and protozoa into living plants. Nematodes are microscopic roundworms that inhabit soil in enormous numbers. Some species feed on bacteria, others on fungi, and some are predators that consume other nematodes. Their feeding activities release nutrients locked in microbial biomass, making them another essential component of the decomposition and nutrient cycling system. Earthworms, perhaps the most familiar soil inhabitants, dramatically alter soil structure as they move through it. They consume soil and organic matter, digesting and depositing it as nutrient-rich castings. Their burrows create channels that allow air and water to penetrate deeper into the soil, improving conditions for root growth and microbial activity. The soil food web connects all these organisms in complex relationships. Energy flows from decomposing organic matter through bacteria and fungi to protozoa and nematodes, then to larger organisms like earthworms and beetles. Each organism contributes to the breakdown and transformation process that keeps nutrients cycling through the living world. This hidden ecosystem beneath our feet operates continuously, day and night, season after season. It is why forests can regenerate after fire, why compost transforms into rich humus, and why plants can grow at all. Without the hidden life of soil, the visible world of plants and animals could not exist. Bacteria are the unseen foundation of soil fertility. These microscopic single-celled organisms are so small that 1 million of them could fit comfortably on the head of a pin. Yet in terms of sheer numbers and ecological importance, they rival any visible form of life on Earth. A fertile soil contains an almost incomprehensible abundance of bacteria. Research has shown that a single gram of healthy agricultural soil may contain as many as 100 million to 1 billion bacterial cells. In forest soils, where organic matter accumulates more densely, numbers can be even higher. This density means that bacteria are the primary agents of decomposition, handling the initial breakdown of the easiest organic compounds. Different bacterial species specialize in different aspects of decomposition. Some break down simple sugars like glucose and fructose, carbohydrates that leak from plant roots or escape from dying cells. Others consume amino acids, the building blocks of proteins, which become available when plant and animal tissues die. Still others target lipids, the fatty compounds found in plant membranes and animal tissues. The metabolic diversity of bacteria is remarkable. Different species produce different enzymes, the biological catalysts that drive chemical reactions in living cells. One bacterial species might produce enzymes that break cellulose, while another specializes in breaking down chitin, the tough material that forms insect exoskeletons and fungal cell walls. Thousands of bacterial species can decompose virtually any organic compound found in nature. Beyond decomposition, many soil bacteria play essential roles in nutrient cycling through nitrogen fixation. Certain bacteria, including species in the genera Azotobacter, Clostridium, and Rhizobium, can extract nitrogen gas from the atmosphere and convert it into ammonia, a form that plants can use. This process, called biological nitrogen fixation, adds significant amounts of nitrogen to ecosystems without any human intervention. Rhizobia are nitrogen-fixing bacteria that form specialized associations with legume plants. They infect legume root hairs and form nodules, where they convert atmospheric nitrogen into ammonia. The plant supplies the bacteria with carbohydrates as energy, while the bacteria provide the plant with nitrogen in a form it can immediately use. This partnership allows legumes to thrive in soils where nitrogen is otherwise scarce. Other bacteria participate in the nitrification process, converting ammonium into nitrate, another plant-available form of nitrogen. Nitrosomonas bacteria convert ammonium to nitrite, and Nitrobacter bacteria convert nitrite to nitrate. This two-step process is essential for making nitrogen available to most plants, though excessive nitrification can contribute to environmental problems when nitrate leaches into waterways. Denitrifying bacteria perform the reverse process under oxygen-free conditions, converting nitrate back into nitrogen gas. This denitrification returns nitrogen to the atmosphere, completing the nitrogen cycle. Wetland soils and waterlogged soils are particularly favorable for denitrification because oxygen diffuses slowly into saturated soils, creating conditions where denitrifying bacteria can actively operate. Bacteria also contribute to soil structure through the production of extracellular polymeric substances. As bacteria grow and metabolize, they release sticky compounds that bind soil particles together into microaggregates. These aggregates, in turn, contribute to soil porosity and water infiltration. Bacterial biomass itself becomes part of the soil organic matter, adding to the living component of soil that sustains its fertility. The bacterial community in soil is not static. It responds to environmental conditions, changing with temperature, moisture, and the availability of different organic compounds. In summer, when soils are warm and microbes are active, bacterial populations flourish. In winter, when temperatures drop, bacterial activity slows, though populations may persist in dormant states waiting for favorable conditions to return. The health of soil bacterial communities reflects the health of the ecosystem above. Soils rich in organic matter from diverse plant communities support more diverse bacterial populations than soils receiving uniform inputs. This relationship works in both directions. Bacterial diversity contributes to plant community diversity by making a wider range of nutrients available. Understanding bacteria is essential for understanding soil fertility. These microscopic organisms are not merely decomposers recycling dead material. They are active participants in ecosystem function, driving the biochemical processes that sustain all terrestrial life. The fungal kingdom includes some of the largest and oldest organisms on Earth. A single honey fungus in Oregon spans over 2,385 acres and is estimated to be around 2,400 years old. While most soil fungi are far more modest in size, they are no less important in their ecological roles. Fungi are indispensable decomposers, symbionts, and ecosystem engineers. Fungi differ fundamentally from plants in their nutritional strategy. Plants produce their own food through photosynthesis, converting sunlight, water, and carbon dioxide into carbohydrates. Fungi, like animals, must obtain their food from external sources. But where animals ingest food and digest it internally, fungi digest their food externally by secreting enzymes into their environment and absorbing the resulting dissolved nutrients. This external digestion makes fungi particularly well suited for decomposing tough plant materials. Cellulose, the primary component of plant cell walls, is a rigid polymer that many bacteria struggle to break down efficiently. Fungi produce cellulases, enzymes that hydrolyze cellulose into glucose molecules that the fungus can absorb. Some fungi also produce lignin peroxidases, enzymes capable of breaking down lignin, the complex aromatic polymer that gives wood its rigidity and resistance to decay. The ecological significance of lignin decomposition cannot be overstated. Lignin is one of the most abundant organic compounds on Earth, accounting for roughly 30% of plant dry mass in woody plants. Only fungi can efficiently break down lignin, making them essential for closing the carbon cycle between plants and the atmosphere. Without fungal decomposition, atmospheric carbon would gradually accumulate in undecomposed plant material. Mushrooms are the fruiting bodies of certain fungi, temporary structures produced for spore dispersal. The main body of the fungus, called the mycelium, grows through soil or decaying wood, exploring for nutrients. When conditions are favorable, usually in response to moisture and temperature cues, the fungus produces mushrooms that release spores into the air. Each mushroom can release billions of spores, theoretically allowing the fungus to colonize new areas. Mycorrhizal fungi form essential associations with plant roots. These fungi infect plant root systems and extend far beyond the root zone, exploring a volume of soil that roots alone could never reach. The mycelium network captures phosphorus, nitrogen, micronutrients, and water, transporting them back to the plant in exchange for carbohydrates produced through photosynthesis. This exchange is so beneficial that over 90% of all plant species form mycorrhizal associations. Different plants form associations with different mycorrhizal fungi. Pine trees, for example, form relationships with certain fungal species that produce edible mushrooms like matsutake in Japan and chanterelles in Europe. Oak trees associate with different fungal species, producing porcini in Italy and truffle-like fungi in California. These specific associations explain why certain mushrooms appear predictably under particular tree species. The mycelium network in forest soils connects trees in ways that scientists are only beginning to understand. Through these fungal networks, carbon, phosphorus, and even defensive chemical signals can move between plants. A paper birch connected to a Douglas fir through shared mycorrhizal fungi can transfer carbon to its neighbor. This underground transfer network has been dubbed the Wood Wide Web by researchers studying forest ecology. Fungal diversity in soil reflects ecosystem health. Diverse plant communities support diverse fungal communities because different plants form associations with different fungal species. Monoculture agriculture, which replaces diverse plant communities with single crops, dramatically reduces fungal diversity. This loss contributes to declining soil health in agricultural systems that rely increasingly on synthetic fertilizers to replace the ecosystem services that fungi once provided. Some fungi are pathogens that attack plant roots, causing diseases like root rot and wilt. These pathogenic fungi play important roles in natural ecosystems by killing susceptible plants and creating gaps in the canopy that allow new species to establish. In agricultural settings, however, pathogenic fungi can cause significant crop losses, requiring human intervention through fungicides or resistant varieties. Fungi also serve as food sources for soil animals. Springtails, mites, nematodes, and other small invertebrates consume fungal hyphae, transferring the nutrients captured by fungi up to larger animals in the soil food web. Some beetles specialize in eating fungi, with different species feeding on different fungal foods. The diversity of fungal feeding animals contributes to the overall diversity of soil communities. The fungal kingdom represents a crucial link between decomposition and ecosystem nutrition. By breaking down tough plant materials that bacteria cannot handle, fungi release nutrients locked in plant tissues back into the soil where new plants can use them. By forming mycorrhizal associations with plant roots, fungi provide the majority of land plants with access to nutrients they could not obtain otherwise. Between the microbial world of bacteria and fungi and the visible world of earthworms and beetles exists a realm of slightly larger soil organisms that are nevertheless difficult to see without magnification. Protozoa and nematodes are the link between microscopic decomposition and larger soil animals. Though individually microscopic, their combined activities drive essential nutrient cycling processes. Protozoa are single-celled eukaryotic organisms that inhabit soil water films. They are animals in the technical sense, meaning they obtain nutrition by consuming other organisms, rather than by photosynthesis like plants or external digestion like fungi. The three main groups of soil protozoa are ciliates, flagellates, and amoebae, each with different feeding strategies and ecological roles. Ciliates are protozoa covered in tiny hair-like structures called cilia that they use for movement and feeding. In soil, ciliates are most common in wet areas and are particularly abundant in soils that are saturated or waterlogged. They feed on bacteria, consuming many bacterial cells each day. As they digest bacteria, they release nutrient waste products that plants can absorb. Flagellates are protozoa that move using whip-like appendages called flagella. They are the most numerous protozoa in soil because they can inhabit even the thin water films that coat soil particles. Many flagellates feed on bacteria, while others eat fungi or other protozoa. Their small size allows them to navigate the maze of spaces between soil particles, accessing bacterial populations and micropores that larger organisms cannot reach. Amoebae move by flowing cytoplasm into extending projections called pseudopodia. They engulf bacterial cells whole through a process called phagocytosis. Some soil amoebae are relatively large, up to 0.5 millimeter in diameter, and can be seen with a hand lens. Testate amoebae construct protective shells from collected particles, which they carry with them as they move through soil. The ecological importance of protozoa lies in their role as bacterial predators. When protozoa consume bacteria, they digest the bacterial biomass and release nutrients contained in those cells. The nutrients are released in forms that plants can readily absorb, primarily as ammonium and other simple nitrogen compounds. This process, called microbial grazing, is one of the primary mechanisms that makes bacterial nutrients available to plants. Research has shown that protozoan grazing can increase plant growth by 60% or more, compared to plants growing in soil where protozoa have been removed. The mechanism is not simply that protozoa make nutrients available, though that is important. Protozoa also stimulate bacterial activity, causing bacteria to grow more rapidly, and cycle nutrients more quickly. The presence of predators activates prey populations, a phenomenon ecologists call the priming effect. Nematodes are microscopic roundworms that are among the most abundant multicellular animals on Earth. A single square meter of grassland soil may contain more than 1 million nematodes, and in forest soils, nematodes are often the most abundant multicellular animals. They range in size from less than a millimeter to several millimeters in length, making them visible with a hand lens but requiring magnification to see in detail. Soil nematodes occupy several different trophic levels in the soil food web. Bacterial-feeding nematodes consume bacteria, ingesting bacterial cells through a food tube in their body. Fungal-feeding nematodes consume fungal hyphae, puncturing fungal cells and ingesting their contents. Other nematodes feed on plants, penetrating plant roots and feeding on internal tissues. Still others are predators, feeding on other nematodes and small soil animals. Like protozoa, nematodes contribute to nutrient cycling by releasing nutrients from their prey. When nematodes consume bacteria or fungi, they release excess nitrogen as ammonium through their body walls. This ammonium diffuses into the soil water and becomes available for plant uptake. Predatory nematodes that eat other nematodes also release nutrients, but from a higher trophic level. Nematodes serve as indicators of soil health because different species are sensitive to different environmental conditions. Some nematodes thrive in compacted soils with limited oxygen, while others require well-aerated conditions. Some tolerate high levels of disturbance, while others are sensitive indicators of soil quality. The nematode community structure reflects the overall health of the soil ecosystem. The combined activities of protozoa and nematodes create what ecologists call a fast carbon cycle in soil. Bacterial and fungal biomass is consumed by these micropredators, and nutrients are released in plant-available forms. This fast cycle contrasts with the slow carbon cycle in which carbon remains in soil organic matter for years or decades before becoming available again. In healthy soils, protozoa and nematodes are abundant and active, driving rapid nutrient cycling that supports vigorous plant growth. In degraded soils, these organisms may be absent or suppressed, contributing to poor nutrient cycling and reduced plant productivity. Their presence and activity are indicators of soil ecosystem health. These microscopic grazers transform the work of decomposers into plant-available nutrients. Without protozoa and nematodes, nutrients locked in bacterial and fungal biomass would remain inaccessible to plants. They complete the link between decomposition and plant nutrition, completing the cycle that sustains terrestrial ecosystems. Earthworms are among the most influential soil animals, though their importance was not recognized until Charles Darwin's extensive research in the late 1800s. Darwin estimated that earthworms could turn over the topsoil of a field in about 50 years, a process he calculated could move as much as 18 tons of soil per acre annually. Modern research has confirmed and refined Darwin's observations, demonstrating that earthworms are fundamental architects of soil structure. Earthworms are segmented worms belonging to the class Oligochaeta, which means few bristles. Unlike their relatives, the polychaete worms, which have many bristles on each segment, oligochaetes have relatively few. There are over 6,000 species of earthworms worldwide, ranging from species barely a centimeter long to giants in the tropics that can reach 3 meters in length. The common name earthworm actually refers to several different ecological groups of worms. Epigeic species live in the surface layer of soil, in leaf litter, and the uppermost centimeters of soil. They are typically small, darkly colored, and adapted to feed on decaying organic matter on the soil surface. Anisic species are larger, burrowing worms that create deep vertical burrows and pull surface organic matter down into their tunnels. Endogeic species live entirely in the soil, creating horizontal burrows as they consume soil and digest its organic content. Earthworm digestion is a remarkable process. Earthworms consume soil through their muscular pharynx, passing it through a muscular gizzard where grinding stones in intentionally swallowed by the worm, help break apart soil particles. The material then passes through the intestine, where digestive enzymes break down organic matter. What emerges as castings is soil with organic matter decomposed, nutrients transformed, and particle size reduced. The casting process has several important effects on soil. Earthworm castings contain higher concentrations of available phosphorus, potassium, calcium, and nitrogen than the surrounding soil. Castings also have higher microbial activity, providing a ready source of decomposers that continues to process organic matter even after passage through the worm. When castings accumulate on the soil surface, they create patches of enriched soil that are focal points for plant germination and growth. Earthworm burrows dramatically alter soil physical properties. Burrows create channels that allow air and water to penetrate more deeply than they could through intact soil. Research has shown that earthworm burrows can increase water infiltration rates by an order of magnitude or more compared to unburrowed soil. This improved infiltration reduces surface runoff and erosion, contributing to water conservation and soil retention. The burrowing action also improves soil aeration. Oxygen diffuses into earthworm burrows more readily than into compacted soil, supporting aerobic microbial activity in the burrow walls. Carbon dioxide produced by soil organisms diffuses out through the same channels. This improved gas exchange supports root respiration in deeper soil layers and enables aerobic decomposition processes that would not occur in compacted, poorly aerated soils. Earthworms are ecosystem engineers that create habitat for other organisms. Their burrows provide shelter for roots, nematodes, mites, springtails, and other small animals. Fungal hyphae grow along burrow walls, exploring the channels created by worm movement, creating biofilm communities that contribute to biochemical processes throughout the soil profile. The relationship between earthworms and plants is complex and bidirectional. Earthworms stimulate plant growth through improved nutrient availability, improved soil structure, and enhanced microbial activity. But earthworms also consume seedlings and fine roots, effects that can be negative for plant establishment in some circumstances. The net effect depends on earthworm abundance, plant species, and soil conditions. Earthworm activity varies dramatically with environmental conditions. Earthworms are most active in warm, moist soils. In temperate regions, earthworm activity peaks in spring and fall when soils are moist and temperatures are moderate. In summer, when soils dry out, earthworms may enter a dormant state, retreating deep into the soil or sealing their burrow openings. In winter, earthworms may also become dormant as temperatures drop below their tolerance range. Earthworms are sensitive to soil disturbance, particularly tillage and pesticide applications. In agricultural soils, earthworm populations are typically much lower than in natural ecosystems because of repeated tillage, pesticide exposure, and crop residue management that reduces organic matter inputs. This reduction in earthworm activity contributes to declining soil structure in agricultural systems. The absence of earthworms in degraded soils is both a symptom and a cause of poor soil health. Restoring earthworm populations is one of the goals of regenerative agriculture practices that minimize tillage, maintain surface residues, and build soil organic matter. Earthworm presence is often used as an indicator of soil recovery in restoration programs. The earthworm's role in soil creation is ongoing and essential. Through their feeding, burrowing, and casting activities, earthworms continuously transform soil, creating structure, improving drainage and aeration, and making nutrients available to plants. They are among the most important animals in terrestrial ecosystems, working continuously beneath our feet to create the soil that sustains the visible world. Soil mites are among the most abundant and diverse arthropods in soil ecosystems. Though most are smaller than a millimeter and go unnoticed by casual observation, they play crucial roles in decomposition, nutrient cycling, and the creation of soil structure. Mites are estimated to constitute 10% or more of the total animal biomass in many soils, making them significant components of below-ground communities. Mites are arachnids, relatives of spiders and scorpions, with 8 legs as adults. Unlike spiders, however, mites have not segmented bodies, and their bodies appear as a single unit. There are over 40,000 described mite species, and researchers estimate that the true number may exceed 1 million. Soil mites occupy several different ecological niches, with different species feeding on fungi, bacteria, dead organic matter, other small invertebrates, or prey. Orobatid mites, also called box mites or armored mites, are among the most numerous soil mites in forest and grassland ecosystems. Their name comes from their ability to roll into a ball, protecting their legs and appendages inside a hardened exoskeleton. They feed primarily on fungi, puncturing fungal hyphae and consuming the internal contents. Some orobatid mites also consume lichen and algae growing on the soil surface. The feeding activities of orobatid mites contribute to fungal community dynamics. By consuming fungal hyphae, mites release nutrients locked in fungal biomass and stimulate fungal regrowth. This grazing pressure maintains fungal populations in a state of active turnover, preventing any single fungal species from dominating. The diversity of orobatid mite species reflects fungal diversity, with different mite species specializing on different fungal foods. Mesostigmatid mites are predators that feed on other small invertebrates, including nematodes, springtails, and other mites. They are larger than most orobatid mites and can be recognized by their large pincer-like chelicerae. Mesostigmatid mites hunt by sensing vibrations and chemical cues, pursuing prey through the soil pore spaces. Their feeding helps regulate populations of smaller soil animals. Prostigmatid mites include several groups with different feeding habits. Some are predators of nematodes and other small invertebrates. Others feed on plant roots, potentially causing damage in agricultural settings. Some prostigmatid mites are fungal feeders, consuming fungal spores and hyphae. Their diversity reflects the diverse resources available in soil microhabitats. Astigmatid mites are soft-bodied mites that often exploit temporary habitats. They are typically found in decaying organic matter, dung, and stored products rather than in mineral soil. Many astigmatid mites have dispersing stages that attach to insects, allowing them to colonize new habitats. Their role in decomposition focuses on organic-rich substrates, like leaf litter and animal remains. Mite abundance and diversity depend on environmental conditions and resource availability. Soils with high organic matter and diverse plant communities support the most diverse mite communities because different plants and plant residues support different fungal and bacterial communities, which in turn support different mite species. Agricultural soils typically have lower mite diversity than natural ecosystems. Mites contribute to soil structure through their feeding and movement activities. The digestion of fungal hyphae by orobatid mites produces fecal pellets that contribute to soil aggregation. The passage of material through the mite gut modifies particle surfaces in ways that affect water holding capacity and nutrient availability. The burrowing and movement of mites creates microchannels that contribute to soil porosity. The relationships between mites and other soil organisms are complex. Mites consume fungi but also disperse fungal spores through their digestive systems. Spores that pass through mite guts often have higher germination rates than unconsumed spores, suggesting that mites may facilitate fungal reproduction. Mites also consume nematodes and other prey, transferring nutrients from those organisms up the soil food web. Springtails are closely related to mites and occupy similar ecological niches. Like mites, springtails are among the most abundant soil arthropods, with some soils containing tens of thousands of individuals per square meter. Their name comes from a forked structure under their abdomen that allows them to spring away from predators. Springtails feed on fungi, bacteria, dead organic matter, and in some cases living plant material. The combined activities of mites and springtails make significant contributions to decomposition and nutrient cycling. By consuming fungal and bacterial biomass, they release nutrients in plant-available forms, similar to the way protozoa and nematodes release nutrients from their prey. Their grazing pressure maintains microbial communities in states of active turnover, stimulating decomposition rates overall. These small arthropods are essential components of soil communities. Their activities connect the microscopic world of bacteria and fungi with the larger world of earthworms and beetles. Without mites and springtails, soil nutrient cycling would proceed more slowly, and the transfer of energy from decomposing organic matter to larger animals would be less efficient. Soil is not a static substrate, but a dynamic system shaped by physical, chemical, and biological processes. Water, air, minerals, and organic matter interact in complex ways that determine soil structure, porosity, and fertility. Understanding these physical properties helps explain why healthy soils support abundant life and how soil degradation occurs. Soil is composed of solid particles and the pore spaces between them. The solid fraction includes mineral particles of different sizes and organic matter in various stages of decomposition. The pore spaces are filled with water, air, or combinations, depending on moisture conditions. The ratio of solids to pores and the distribution of pore sizes determines how well soil stores and transmits water and air. Mineral particles in soil range from clay particles smaller than 0.002 millimeters in diameter to stones and boulders larger than 250 millimeters. Between these extremes, sand particles range from 0.05 to 2 millimeters, and silt particles range from 0.002 to 0.05 millimeters. The relative proportions of these different sized particles determine soil texture, a property that strongly influences water retention and drainage. Sandy soils have large pores that drain quickly but hold little water. Clay soils have small pores that hold more water but drain slowly, sometimes so slowly that plant roots suffocate from lack of oxygen. Loamy soils have a balanced mixture of particle sizes that provides both drainage and water retention, making them the most productive soils for plant growth. Soil structure refers to how mineral particles and organic matter are aggregated into larger units called aggregates or PEDs. Aggregates form through the combined action of plant roots, fungal hyphae, earthworm activity, and microbial products that bind particles together. Well-aggregated soil has stable structure that resists erosion and maintains porosity even under rainfall impact or tillage disturbance. Biological activity is fundamental to aggregate formation and stability. Fungal hyphae physically enmesh soil particles, creating aggregates that hold together. Bacterial polysaccharides and fungal glomalin, a glycoprotein produced by mycorrhizal fungi, act as glues that bind particles together. Earthworm mucus and casts contribute additional binding materials. The decay of dead roots and hyphae leaves behind organic residues that maintain aggregate stability over time. Soil porosity consists of pores of different sizes, serving different functions. Large pores greater than 0.05 millimeters in diameter drain rapidly after rainfall and provide pathways for root penetration and soil fauna movement. Medium pores between 0.005 and 0.05 millimeters hold water available for plant uptake. Small pores less than 0.005 millimeters hold water too tightly for plant roots to extract. The balance of water and air in soil pores is determined by moisture content. When soil is saturated, all pores are filled with water. As water drains from large pores, air enters and fills the vacated spaces. This cycling of water and air is essential. Roots need oxygen for respiration, while plant-available water resides in the pore spaces that drain more slowly. Soil organic matter is the fraction of soil composed of decaying plant and animal residues in various stages of decomposition. It ranges from readily decomposable fresh residues to highly transformed humus that may persist for centuries. Organic matter influences soil physical properties by improving aggregation, increasing water holding capacity, and darkening soil color. The decomposition of organic matter is mediated by soil organisms. Bacteria and fungi decompose simple compounds, releasing carbon dioxide and nutrients. Their biomass becomes part of the soil organic matter pool. The bodies of dead organisms contribute to organic matter at various stages of transformation. Earthworms and other soil animals consume organic matter and accelerate its decomposition. Humus, the stable end product of decomposition, is resistant to further decay and can persist in soil for thousands of years. It contributes to soil fertility by holding nutrients in forms exchangeable with the soil solution, improving soil aggregation, and increasing water holding capacity. The dark color of humus-rich soils also increases heat absorption, advancing spring warming. Soil temperature is influenced by many factors including color, moisture content, and vegetation cover. Dark soils absorb more solar radiation than light soils. Moist soils require more energy to warm than dry soils because water has a high heat capacity. Vegetation moderates temperature by shade and by influencing snow cover in winter. Freeze-thaw cycles affect soil structure through the mechanical disruption of aggregates. When water in soil freezes, it expands by about 9%, pushing soil particles apart. When ice melts, the expanded pore spaces remain, temporarily increasing porosity. Repeated freeze-thaw cycles can break apart compacted soil, improving structure in some cases, but causing heaving damage to plant roots in others. The physical properties of soil are not static, but change constantly in response to biological activity, weather, and human management. Soils under perennial vegetation maintain stable structure through continuous root growth and fungal activity. Tilled agricultural soils experience regular structure disruption that can lead to compaction and degradation over time. Understanding these dynamics is essential for managing soils sustainably. Soil chemistry determines which nutrients are available to plants and how they cycle through ecosystems. The mineral component of soil provides most of the nutrients that plants need, but only certain forms are accessible to roots. Understanding soil chemistry helps explain why some soils are fertile while others are impoverished, and how human activities can alter nutrient availability. Most soil nutrients come from mineral weathering, the gradual breakdown of primary minerals by water, oxygen, carbon dioxide, and organic acids. Feldspars, micas, and apatites release potassium, calcium, magnesium, and phosphorus as they weather. The rate of weathering depends on mineral type, surface area exposed to weather, temperature, and moisture. In young soils, primary minerals dominate nutrient supply. In older soils, they may be largely depleted. Cation exchange capacity is a measure of a soil's ability to hold and release positively charged nutrient ions. Clay particles and organic matter carry negative charges that attract positively charged ions like calcium, magnesium, potassium, and ammonium. These nutrients adhere to soil particles, but can be released into the soil solution where roots can absorb them. Soils with high clay and organic matter content have higher cation exchange capacity and greater nutrient holding ability. Base saturation refers to the proportion of cation exchange sites occupied by basic nutrient cations, primarily calcium, magnesium, and potassium, versus acidic cations like hydrogen and aluminum. Soils with high base saturation are fertile because they hold abundant nutrients in plant-available forms. Soils with low base saturation are acidic and may have aluminum toxicity problems that limit plant growth. Soil pH measures the concentration of hydrogen ions in soil solution on a scale of 0 to 14, where 7 is neutral. Values below 7 are acidic, above 7 are alkaline. Most plants grow best at slightly acidic to neutral pH between 6 and 7, where most nutrients are most available. At very low pH, aluminum becomes soluble and toxic to roots. At high pH, iron, manganese, and phosphorus become less available. Nutrient availability is strongly pH dependent. Phosphorus is most available between pH 6 and 7, where it forms soluble compounds with calcium and iron. Outside this range, phosphorus reacts with aluminum or calcium to form insoluble compounds that plants cannot access. Liming acidic soils and acidifying alkaline soils can improve phosphorus availability. Nitrogen is unique among major nutrients because it cycles through organic matter before becoming available to plants. Organic nitrogen in decomposing matter must be converted to ammonium or nitrate by soil microbes before roots can absorb it. This mineralization process depends on microbial activity, which is influenced by temperature, moisture, and carbon availability. Synthetic fertilizers bypass this cycling, providing nitrogen in immediately available forms. Phosphorus is essential for energy transfer in plants, but it is among the least mobile nutrients in soil. Most phosphorus is bound to soil particles or calcium compounds and becomes available very slowly. Plant roots acquire phosphorus mainly by exploring new soil volumes, releasing phosphatase enzymes that release phosphate from organic compounds. Mycorrhizal fungi dramatically extend the volume of soil that roots can explore. Potassium is essential for enzyme activation and water regulation in plants. It is held on cation exchange sites and moves to roots by diffusion through the soil solution, a slow process that requires adequate soil moisture. Potassium deficiency is common in sandy soils that have low cation exchange capacity and in heavily crocked fields where potassium removal exceeds replacement. Micronutrients including iron, manganese, zinc, copper, boron, and molybdenum are required in small quantities, but are no less essential than macronutrients. Their availability varies with pH and oxidation reduction conditions. In alkaline soils, iron and manganese become less available, causing deficiency symptoms in plants that require them. In acidic soils, they may become too available, causing toxicity. Redox potential describes whether soil conditions are oxidizing or reducing. In well-aerated soils, oxygen is abundant and conditions are oxidizing. In waterlogged soils, oxygen becomes depleted and anaerobic processes dominate. Under reducing conditions, iron and manganese change from oxidized forms that are relatively insoluble to reduced forms that are more soluble and sometimes toxic. Anions, negatively charged nutrient ions like nitrate, phosphate, and sulfate, behave differently than cations in soil. They are not held on exchange sites but move with soil water and may leach from root zones. Nitrate is particularly prone to leaching because it does not bind to soil particles. Phosphite and organic forms of phosphorus are less mobile but may not be available to plants. Soil chemistry is dynamic, responding to management practices and environmental conditions. Fertilizer applications alter nutrient balances. Irrigation changes moisture regimes that affect nutrient movement. Tillage exposes new mineral surfaces to weathering. These changes can improve or degrade soil fertility over time, depending on management practices. The nitrogen cycle is one of the most important nutrient cycles in soil ecosystems. Nitrogen is a component of proteins, nucleic acids, and chlorophyll, making it essential for all living organisms. The atmosphere is 78% nitrogen, but this gaseous nitrogen is unavailable to most organisms. Only certain bacteria can convert atmospheric nitrogen into forms that other organisms can use, creating a dependency that links all life to microbial processes. Biological nitrogen fixation is the process by which certain bacteria convert atmospheric nitrogen gas into ammonia. These bacteria contain the enzyme nitrogenase, capable of breaking the strong triple bond between nitrogen atoms. The reaction requires substantial energy, which the bacteria obtain from carbohydrates supplied by plant partners or from organic compounds in their environment. Fixation converts inert atmospheric nitrogen into biologically useful ammonia. Free-living nitrogen-fixing bacteria include species in the genera Azotobacter and Clostridium. Azotobacter are aerobic bacteria that fix nitrogen in soil and rhizosphere, providing nitrogen to associated plants through root exudates. Clostridium are anaerobic bacteria that fix nitrogen in waterlogged soils where oxygen is limited. Cyanobacteria, formerly called blue-green algae, fix nitrogen in aquatic environments and in soil crusts in arid regions. Symbiotic nitrogen-fixing bacteria form associations with plant roots. Rhizobia infect legume root hairs, forming characteristic nodule structures where nitrogen fixation occurs. Inside nodules, the bacteria differentiate into bacteroids that cannot revert to free-living forms but that fix nitrogen continuously. The plant supplies carbohydrates to the bacteroids and receives ammonia in return, a mutually beneficial exchange that allows legumes to thrive in nitrogen-poor soils. The legume-rhizobia symbiosis is one of the most efficient nitrogen-fixing relationships known. Under ideal conditions, fixing over 10 pounds of nitrogen per acre per year, equivalent to over 200 pounds of ammonium sulfate fertilizer. Different legume species associate with different rhizobial species, and specific strain-plant combinations can vary dramatically in fixing efficiency. Ammonification is the process by which decomposer organisms convert organic nitrogen in dead matter into ammonia. Proteins, nucleic acids, and other organic nitrogen compounds are broken down by bacteria and fungi, releasing ammonium as a byproduct. This ammonium can be absorbed by plant roots or converted by other bacteria into nitrate. Ammonification is continuous wherever decomposition occurs, recycling organic nitrogen back into inorganic forms. Nitrification is the two-step conversion of ammonia to nitrate. First, Nitrosomonas bacteria convert ammonia to nitrite. Then Nitrobacter bacteria convert nitrite to nitrate. Both steps require oxygen and are inhibited by acidic conditions. Nitrate is the preferred nitrogen form for most plants, diffusing more readily to roots than ammonium and causing less pH change in the rhizosphere. Denitrification is the anaerobic conversion of nitrate back to nitrogen gas. Denitrifying bacteria use nitrate as an electron acceptor instead of oxygen when oxygen is scarce, as in waterlogged soils or in anaerobic microsites within otherwise well-aerated soil. The process releases nitrogen back to the atmosphere, completing the cycle. Wetland rice fields and riparian zones are particularly important denitrification sites. Ammonium fixation occurs when ammonium ions become trapped between layers of certain clay minerals. Fixed ammonium is not available to plants, but may be slowly released as the soil weathers. Illite and vermiculite clays fix significant amounts of ammonium, while kaolinite fixes little. Soils with high levels of ammonium-fixing clays may require more nitrogen fertilizer to achieve the same plant growth. Leaching is the downward movement of nitrate with percolating water. Because nitrate is not held on negatively charged soil particles, it moves freely with soil water. In high rainfall areas or with excessive irrigation, nitrate can leach below the root zone, into groundwater, or into surface waters. Leaching is a significant source of water pollution and of nitrogen loss from agricultural systems. The nitrogen cycle has been dramatically altered by human activities. The Haber-Bosch process, developed in the early 20th century, converts atmospheric nitrogen to ammonia for fertilizer production. This industrial fixation now exceeds biological fixation, providing the nitrogen needed to grow high-yielding crops. However, excess nitrogen from fertilizer and manure applications frequently leaches into waterways, causing eutrophication. Managing the nitrogen cycle in agricultural systems requires balancing supply with crop demand. Too little nitrogen limits yields, too much nitrogen wastes resources and pollutes waters. Timing applications to match crop uptake, using slow-release forms, and including cover crops that capture nitrogen can all improve nitrogen use efficiency. Understanding the biological processes driving the cycle is essential for sustainable nitrogen management. The carbon cycle in soil connects the atmosphere, living organisms, and geological reservoirs in a continuous exchange that shapes global climate. Soil is the largest terrestrial reservoir of carbon, holding more carbon than the atmosphere and all living vegetation combined. The balance between carbon inputs to soil through decomposition and carbon losses through respiration and leaching determines whether soil acts as a carbon sink or source. Organic carbon enters soil through plant residues, animal remains, and root exudates. Leaves, stems, and roots that die each year contribute organic matter that decomposers process. Living roots release carbohydrates, amino acids, and other compounds that feed soil microbes. These root exudates can contribute 20% or more of the carbon allocated to below-ground productivity in some plants. Decomposition converts organic carbon to carbon dioxide through microbial respiration. Bacteria and fungi consume organic matter, using carbon compounds for energy and releasing carbon dioxide as a byproduct. The rate of decomposition depends on substrate quality, with simple compounds decomposing rapidly and complex compounds like lignin decomposing slowly. Temperature and moisture also influence decomposition rates, with faster decomposition in warm, moist conditions. Soil organic matter persists at different timescales. Labile carbon decomposes in weeks to months, cycling rapidly through microbial biomass. Intermediate carbon may persist for years to decades in slow-cycling pools. Stable carbon, also called humus, is chemically resistant to further decomposition and can persist for centuries or millennia. The proportion of organic matter in each pool determines how long carbon remains in soil. Carbon stabilization mechanisms include chemical recalcitrance, physical protection, and biochemical stabilization. Lignin and related compounds are chemically resistant to microbial attack. Carbon protected within soil aggregates is inaccessible to decomposers. Organic matter bound to mineral surfaces through cationic bridges is protected from decomposition. These mechanisms interact, with most soil carbon protected through multiple mechanisms. Microbial necromass, the carbon in dead microbial cells, contributes significantly to soil organic matter. Fungal hyphae and bacterial cells that die become part of the soil organic carbon pool. The chitin in fungal cell walls and the phospholipids in all cell membranes are particularly resistant to decomposition and contribute to stable carbon pools. Carbon dioxide diffuses from soil to atmosphere through respiration by roots, microbes, and soil animals. This soil respiration is one of the largest fluxes in the global carbon cycle, releasing nearly 10 times as much carbon as fossil fuel combustion. The magnitude of soil respiration varies with temperature, moisture, and substrate availability, responding dynamically to environmental conditions. Methane production occurs in waterlogged soils where anaerobic conditions favor methanogenic bacteria. Wetland soils, flooded rice fields, and hydric soils in general are sources of methane, a greenhouse gas 25 times more potent than carbon dioxide. Methane oxidizing bacteria in oxic surface layers consume some methane before it escapes to the atmosphere, but significant emissions occur from many wetlands. The global soil carbon stock is approximately 1,500 to 2,400 gigatons of carbon, depending on estimates and depth considered. This is 2 to 3 times the amount of carbon in the atmosphere and about 3 times the amount in all living vegetation. Small changes in soil carbon stocks could significantly affect atmospheric carbon dioxide concentrations and global climate. Soil carbon responds to land use and management changes. Converting natural ecosystems to agriculture typically reduces soil carbon, sometimes by 30 to 50%. Tillage accelerates decomposition by breaking aggregates and exposing organic matter to microbial attack. Removing crop residues removes carbon inputs that would otherwise contribute to soil organic matter. Restoring carbon requires building inputs while minimizing losses. Regenerative agriculture practices aim to increase soil carbon. No-till farming reduces disturbance and preserves aggregates. Cover crops provide year-round carbon inputs and living roots that exude carbon. Diverse rotations increase the variety and quantity of carbon inputs. Organic amendments, including compost and manure, add carbon directly. These practices can rebuild soil carbon over time. The potential for soil carbon sequestration is substantial. Some researchers estimate that agricultural soils could sequester several gigatons of carbon per year, equivalent to a significant fraction of anthropogenic emissions. Practical implementation faces challenges including maintaining carbon inputs, accessing land, and verifying sequestration. Climate change affects soil carbon through altered temperature, moisture, and vegetation. Warmer temperatures accelerate decomposition, potentially releasing carbon stored in soils. Changed precipitation patterns affect decomposition and plant productivity. Altered vegetation changes carbon inputs. These feedback effects are complex and uncertain. The soil carbon cycle is central to climate regulation and agricultural productivity. Managing soils to store more carbon addresses both climate change and food security, a rare opportunity where environmental and agricultural goals align. Soil biodiversity encompasses the incredible variety of life in soil, from microscopic bacteria to visible earthworms. This diversity drives the ecosystem processes that sustain all terrestrial life, including decomposition, nutrient cycling, and soil structure formation. Understanding soil biodiversity helps explain why some soils are more productive than others, and how human activities can either support or degrade the life beneath our feet. Bacteria are the most numerous organisms in soil, with cell counts in the millions per gram in most soils and up to a billion per gram in the most fertile soils. Different bacterial species perform different functions. Some decompose specific compounds, others fix nitrogen, and still others form associations with plant roots. Bacterial community composition changes with soil conditions, responding to pH, moisture, carbon availability, and plant community composition. Fungi are as diverse as bacteria in terms of species, though typically less numerous in cell count. Different fungal groups dominate in different ecosystems. Ectomycorrhizal fungi in forests, arbuscular mycorrhizal fungi in grasslands and agricultural fields, and saprophytic fungi that decompose dead organic matter. Fungal hyphae explore soil volumes that bacterial cells cannot access, making fungal activity essential for capturing nutrients from distant sources. Arthropods add macro-level diversity to soil communities. Mites and springtails are abundant in virtually all soils, feeding on fungi, bacteria, dead organic matter, and each other. Their grazing activities stimulate microbial activity and contribute to nutrient cycling. Larger arthropods, including ants, beetles, and termites, modify soil structure through their burrowing and feeding activities. Earthworms are perhaps the most familiar soil animals, but they are just one of several groups of megafauna that influence soil processes. Ants excavate extensive burrow systems that improve drainage and aeration. Termites build mounds that concentrate nutrients and create specialized microhabitats. These large soil animals significantly modify their environment. Species richness in soil is enormous, but poorly documented. Current estimates suggest that a single gram of soil may contain thousands of bacterial species and tens of thousands of fungal species. The total diversity across all soils may exceed tens of millions of species. Most soil organisms are undescribed, and the vast majority have never been cultured in laboratories. Functional diversity matters as much as species diversity. The soil food web includes organisms performing every ecological function: primary decomposition, secondary consumption, predation, nitrogen fixation, nitrification, denitrification, and more. The loss of any functional group can alter ecosystem processes, though resilience often comes from functional redundancy where multiple species can perform the same function. Soil biodiversity supports ecosystem services that humanity depends upon. Decomposition recycles nutrients essential for agriculture. Nutrient cycling makes fertilizers unnecessary in natural ecosystems. Soil aggregation prevents erosion and maintains soil structure that stores water. These services have enormous economic value, though they are rarely accounted for in market transactions. Agricultural practices affect soil biodiversity differently. Monoculture agriculture reduces plant diversity, which in turn reduces fungal and bacterial diversity associated with diverse plant communities. Tillage physically disrupts soil communities, destroying fungal networks and earthworm habitats. Pesticides kill non-target organisms, including beneficial microbes and arthropods. Irrigation and fertilization alter environmental conditions that affect community composition. Organic agriculture supports higher soil biodiversity than conventional agriculture, though yields are often lower. Organic practices including cover cropping, crop rotation, reduced tillage, and organic amendments maintain more diverse soil communities than synthetic inputs and tillage. The trade-offs between biodiversity and productivity depend on management intensity and market conditions. Restoration of degraded soils can rebuild biodiversity over time. Removing disturbance, adding organic matter, and planting diverse communities can gradually restore soil communities. Recovery trajectories depend on soil type, climate, and management history. Some degraded soils may require decades to centuries to fully restore soil biodiversity. Climate change affects soil biodiversity through altered temperature, moisture, and carbon dynamics. Warmer temperatures may increase microbial activity and decomposition, potentially depleting soil carbon. Changed precipitation regimes alter the moisture conditions that many soil organisms require. These changes may reduce soil biodiversity in some regions, while possibly increasing it in others. The connection between soil biodiversity and human health is increasingly recognized. The human gut microbiome includes many of the same bacterial lineages found in soil. Exposure to soil microbes in childhood may be important for immune development. The decline of soil biodiversity in urban environments may contribute to increased autoimmune and allergic diseases. Protecting soil biodiversity requires recognizing its value and managing to support it. Reducing tillage, maintaining organic matter inputs, avoiding broad-spectrum pesticides, and preserving natural areas all contribute to soil biodiversity conservation. The hidden life beneath our feet is both essential and endangered. The phosphorus cycle is unlike other nutrient cycles because it lacks a gaseous phase. Phosphorus moves from rocks through soils and living organisms back to rocks, but it does not enter the atmosphere. This unique chemistry creates different dynamics than nitrogen or carbon cycling, with implications for ecosystem fertility and human management. Most soil phosphorus comes from mineral weathering. Apatite, the primary phosphorus-containing mineral in most rocks, releases phosphate as it weathers. The rate of release depends on mineral type, surface area, temperature, and pH. In young soils derived from phosphorus-rich parent material, weathering supplies adequate phosphorus. In old, highly weathered soils, phosphorus may be depleted. Soil phosphorus exists in several pools that differ in availability. Inorganic phosphorus includes phosphate ions in soil solution and phosphate bound to mineral surfaces. Organic phosphorus is bound in plant and microbial biomass, in dead organic matter, and in humus. Plant-available phosphorus is primarily in solution or loosely bound to iron and aluminum compounds in acidic soils and calcium compounds in alkaline soils. The transformation between pools is mediated by biological and chemical processes. Organic phosphorus is released through microbial decomposition, a process called mineralization. Inorganic phosphorus becomes available through desorption from mineral surfaces and dissolution from solid compounds. These processes are slow compared to plant demand, making phosphorus often limiting to plant growth. Mycorrhizal fungi dramatically improve phosphorus acquisition. The hyphal network extends far beyond the root depletion zone, exploring new soil volumes for phosphorus. Some fungi release organic acids that release phosphate from mineral compounds. Others release phosphatases that release phosphate from organic compounds. The plant supplies carbohydrates to the fungus and receives phosphorus in return. Plant species differ in phosphorus acquisition strategies. Some plants release phosphatases from their roots that release phosphate from organic matter. Others release exudates that solubilize inorganic phosphorus. Cluster roots in some species exude large amounts of organic acids that release phosphate from minerals. These adaptations allow different species to coexist in phosphorus-limited environments. Phosphorus moves to roots by diffusion, a slow process requiring adequate soil moisture. The diffusion coefficient for phosphate is very low because phosphate reacts readily with soil particles. Roots acquire phosphorus by depleting the solution around them, creating a gradient that drives diffusion. Mycorrhizal hyphae extend this depletion zone dramatically. The ratio of carbon to phosphorus in microbial biomass is much tighter than in plant material. When microbes die and decompose, they release more phosphorus than plants can immediately use. This excess phosphorus becomes available for plant uptake, creating a microbial pathway for phosphorus cycling that is faster than chemical release from minerals. Leaching losses of phosphorus are small compared to nitrogen because phosphorus reacts strongly with soil particles. However, eroded soil particles carry phosphorus attached to them, potentially causing eutrophication in downstream waters. In sandy soils with low phosphorus retention, leaching can contribute significant phosphorus losses. Drainage tiles in artificially drained soils can carry phosphorus to ditches and streams. Human activities have dramatically altered the phosphorus cycle. Mining of phosphate rock creates a new source of phosphorus that bypasses the slow weathering cycle. Synthetic fertilizers make phosphorus immediately available, supporting high crop yields, but also causing environmental problems when excess phosphorus reaches waterways. The world phosphate rock resource is finite, though estimates vary dramatically. At current consumption rates, reserves may last from several decades to several centuries. Morocco, China, and the United States hold the largest reserves. The geographic concentration of reserves creates geopolitical dependencies that could affect long-term phosphorus availability. Managing phosphorus sustainably requires closing the loop between agriculture and food production. Recycling phosphorus from human waste, from animal manure, and from food processing byproducts can reduce mining requirements. Reducing phosphorus losses through erosion control and precision fertilizer application can extend existing reserves. The unique chemistry of the phosphorus cycle makes it particularly challenging to manage sustainably. Unlike nitrogen, which can be fixed from the atmosphere, phosphorus must be mined or recycled. Losses are difficult to recover once phosphorus enters waterways. Sustainable phosphorus management requires both reducing inputs and recycling existing phosphorus through food systems. The potassium cycle involves the movement of potassium between soil, plants, and the atmosphere. Unlike nitrogen and carbon, potassium does not enter organic molecules but remains as an ion throughout its cycle. This simple chemistry creates a cycle that is less complex than other nutrient cycles, but presents unique challenges for soil management. Potassium is abundant in most soils, comprising about 2.5% of the Earth's crust. However, only a small fraction is available to plants at any time. Most potassium is locked in mineral structures, released only through slow weathering processes. The balance between rapidly cycling potassium in soil solution and slowly available potassium in minerals determines potassium fertility. The major pools of potassium in soil are solution potassium, exchangeable potassium, non-exchangeable potassium, and mineral potassium. Solution potassium is immediately available, but is present in very small quantities, typically less than 1% of total potassium. Exchangeable potassium is held on clay mineral surfaces and can be released into solution. Non-exchangeable potassium is held between clay mineral layers and is released slowly. Mineral potassium is structurally bound in feldspars and micas and is released through weathering. Potassium in solution is the fraction immediately available to plants. Roots absorb potassium as the potassium ion from soil solution. The concentration in solution is maintained by equilibrium with exchangeable potassium on clay particles. When plants remove solution potassium, exchangeable potassium is released to maintain equilibrium. This buffering protects against sudden changes in potassium availability. The release of non-exchangeable potassium to exchangeable and solution forms depends on the mineral type. Vermiculite and illite clays can hold substantial non-exchangeable potassium. When exchangeable potassium is depleted, the non-exchangeable pool can release potassium to maintain availability. This release is slow, occurring over weeks to months, making non-exchangeable potassium a reserve source rather than an immediate source. Mineral weathering is the ultimate source of potassium in most soils. Feldspars are the primary potassium-containing minerals, releasing potassium as they weather to clay minerals. The rate of release depends on mineral type, particle size, temperature, moisture, and acidity. Feldspar weathering is very slow, measured in centuries to millennia, so mineral potassium is a long-term reserve rather than an immediate source. Potassium uptake by plants removes potassium from the soil system. When crops are harvested, potassium is removed along with the biomass. Potassium cycles in leaf litter and animal waste. In forests, potassium recycles through leaf fall and decomposition. In agricultural systems, harvest removal is the primary loss pathway, typically balanced by fertilizer additions. Leaching losses of potassium are generally small in mineral soils because potassium is held on exchange sites. However, in sandy soils with low cation exchange capacity, potassium can leach significantly. In high rainfall areas or with excessive irrigation, potassium leaching can contribute to deficiencies. In organic soils with low clay content, leaching can be substantial. The potassium to calcium ratio affects plant growth and soil health. High potassium relative to calcium can induce magnesium deficiency. Low potassium relative to calcium can affect cell wall structure and plant water regulation. Balanced nutrition requires attention to ratios between nutrients, not just absolute amounts. Potassium fertilizers include potassium chloride, potassium sulfate, potassium nitrate, and organic sources like green sand. Potassium chloride is the most common source but adds chloride that can accumulate in some soils. Potassium sulfate provides both potassium and sulfur, benefiting crops requiring both nutrients. Organic sources release potassium more slowly and add other benefits. The potassium cycle is simpler than nitrogen or phosphorus cycles because it lacks biological fixation. However, managing potassium fertility remains challenging because the cycling between pools is slow. Adequate potassium availability requires managing both inputs and losses to maintain the balance between immediately available and reserve pools. Deficiency symptoms include reduced growth, leaf curling, and marginal chlorosis. Excess potassium interferes with magnesium uptake, causing deficiency symptoms, even when magnesium is adequate. Both deficiency and excess can limit plant productivity. The sulfur cycle shares some characteristics with both nitrogen and phosphorus cycles. Like nitrogen, sulfur is essential for biological molecules and cycles between organic and inorganic forms. Like phosphorus, sulfur ultimately derives from mineral weathering. The combination creates a complex cycle with both biological and geological components that requires careful management. Sulfur enters the atmosphere from several natural sources. Volcanic emissions release sulfur dioxide that can enter the atmosphere. Marine aerosols contain sea salt sulfate that deposits on land. Biological processes produce hydrogen sulfide and dimethyl sulfide from marine and terrestrial ecosystems. The atmosphere is a key pathway for sulfur movement between ecosystems. Deposition brings sulfur from the atmosphere to the surface. Sulfur dioxide dissolves in rainwater, forming sulfuric acid. Particulate sulfate settles through dry deposition. Coastal areas receive sea salt sulfate from sea spray. Background deposition is supplemented by human emissions from coal combustion and other industrial processes. In soil, sulfate is the primary plant available form. Sulfate is negatively charged and is not held on cation exchange sites, so it moves readily in soil water. Plants absorb sulfate and reduce it to sulfide for incorporation into proteins and other sulfur-containing compounds. The reduction requires energy and specialized enzymes. Mineral sulfur is present in some soils as elemental sulfur or as sulfate minerals like gypsum. Elemental sulfur must be oxidized to sulfate by soil bacteria before plants can use it, making elemental sulfur a slow-release source. Gypsum dissolves directly to release sulfate. These mineral sources weather slowly, contributing to long-term sulfur supply. Biological sulfur cycling includes fixation, mineralization, and volatilization. Certain bacteria can fix sulfate, incorporating it into organic compounds. Decomposing organic matter releases sulfate through mineralization. Plants release sulfur gases through their roots, contributing to atmospheric sulfur. These biological processes drive the local sulfur cycle. Organically bound sulfur is the largest pool in most soils. Sulfur is incorporated into proteins as cysteine and methionine. The organic sulfur pool turns over through decomposition. The ratio of carbon to sulfur in organic matter is roughly 100 to 1, meaning organic matter contains about 1% sulfur. Building soil organic matter builds sulfur reserves. Sulfur deficiency is common in agricultural systems because sulfur is not supplied by most fertilizers. High-yielding crops remove significant sulfur in harvested biomass. Atmospheric deposition has decreased due to clean air regulations, reducing this source. The combination has increased sulfur deficiency in many regions. Sulfur functions in plants include protein structure, enzyme activity, and defense compounds. Sulfur is a component of cysteine and methionine, essential amino acids. Sulfur-containing enzymes catalyze important reactions. Sulfur compounds in onions and other plants provide defense against herbivores and pathogens. Deficiency symptoms include chlorosis similar to nitrogen deficiency, but often appearing on younger leaves first. Sulfur does not translocate readily from older to younger leaves, so deficiency appears in new growth. Reduced protein synthesis affects growth rates. Oilseed crops require particularly high sulfur for seed development. Sulfur contamination occurs through industrial emissions and volcanic activity. Sulfur dioxide dissolves in rainwater, forming acid rain that damages forests and lakes. Acidification mobilizes aluminum in soils, causing toxicity to plant roots. Reduced emissions from industrial sources have decreased this source in many regions. Sulfur fertilizers include elemental sulfur, gypsum, ammonium sulfate, and potassium sulfate. Elemental sulfur requires oxidation, providing slow release over multiple years. Gypsum provides immediately available sulfate and also improves soil structure. Ammonium sulfate provides both nitrogen and sulfur. Organic sources like compost release sulfur slowly. The sulfur cycle is managed most easily by attention to inputs and outputs. Atmospheric deposition decreases as industrial sources are controlled. Fertilizer applications must account for crop removal and soil supply. Building organic matter provides a slow-release sulfur source that mineralizes as plants need it. Soil formation is the process by which rock weathers into soil through the combined action of climate, organisms, topography, and time. These 5 factors interact to create the incredible diversity of soil types around the world. Understanding soil formation helps explain why soils differ from place to place and how long soil development takes. Parent material is the unconsolidated material from which soils form. It includes weathered rock fragments on hill slopes, glacial deposits left by ice ages, alluvial sediments deposited by rivers, and wind-blown loess and sand. The composition of parent material determines the starting point for soil development, influencing soil texture, mineralogy, and chemical properties. Climate controls the rate of weathering and the balance between physical, chemical, and biological processes. Warm, moist climates accelerate weathering, leading to rapid soil development. Cold climates slow processes, producing younger soils. Arid climates produce minimal weathering, resulting in poorly developed soils. Precipitation affects the balance between leaching and accumulation. Biological organisms contribute to soil formation through multiple mechanisms. Lichens and mosses colonize bare rock, initiating weathering. Plant roots penetrate rock, wedging it apart physically and releasing organic acids chemically. Earthworms and other mixing organisms blend mineral and organic material. The products of decomposition accumulate as organic matter that contributes to soil development. Topography affects soil formation through its influence on drainage, erosion, and material accumulation. Steep slopes erode faster than soils can develop, maintaining shallow soils. Flat valleys accumulate material, developing deeper soils. Aspect controls solar radiation and moisture balance, creating differences between north and south-facing slopes. Time is required for soil development, with processes measured in centuries to millennia. The oldest soils may have developed continuously for hundreds of thousands of years. The youngest soils form on recent deposits like floodplains and volcanic ash. Most agricultural soils have developed over thousands of years, though some have been modified dramatically by human activities. The soil formation process proceeds through stages. Bare rock weathers initially through physical and chemical processes. Organic matter accumulates from pioneer organisms. Weathering releases nutrients that support more abundant vegetation. Soil organisms mix organic and mineral material. Eventually, recognizable soil horizons develop through these continuous processes. Soil horizons are distinctive layers that form in response to soil-forming processes. The O horizon contains organic matter at various decomposition stages. The A horizon is where mixing of mineral and organic material occurs. The E horizon is where alluviation removes material. The B horizon accumulates materials translocated from above. The C horizon is weathered parent material. Bedrock underlies the profile. Laterization is a soil-forming process in warm, wet climates that leads to iron-rich soils. Intense weathering releases iron and aluminum oxides that accumulate in the subsoil, giving red and yellow colors. Silica is leached away. Lateritic soils are common in the tropics but are often infertile because of nutrient depletion. Podzolization produces ash-gray soils in cool, forest-covered climates. Acids from conifer needles mobilize iron and aluminum, moving them from the surface horizon and depositing them in lower horizons. The surface becomes depleted and bleached, giving characteristic gray colors. Podzols are common in boreal forests and mountain areas. Glaciation produces gray, anaerobic soils in waterlogged conditions. Iron is reduced to the ferrous form, which is mobile and gray. When waterlogged soils dry periodically, iron oxidizes and forms mottles. Glaze are common in wetlands and depressional topography. Calcification produces calcium-rich soils in arid to subhumid climates. Calcium carbonate accumulates as lime nodules or layers as calcium moves with infiltrating water and precipitates as the water evaporates. Calcisols are common in the Great Plains of the United States and in Mediterranean climates around the world. Salinization produces salt-affected soils in arid climates with inadequate drainage. Evaporation draws water upward, carrying dissolved salts that precipitate as the water evaporates. Saline soils have white salt crust on the surface. Alkali soils have high sodium that disperses clay and destroys soil structure. Both are problems in irrigated agriculture. Human activities have dramatically accelerated or altered soil-forming processes. Agriculture accelerates erosion beyond natural rates. Irrigation alters water balance and can cause salinization. Deforestation alters biological inputs. Urbanization removes soils entirely. These changes have created novel conditions that natural soil-forming processes cannot address. Understanding soil formation helps guide soil management. Soils in early stages of development require different management than mature soils. Climate determines the rate of processes like organic matter decomposition. Topography affects drainage and erosion vulnerability. The history of soil formation affects the resilience of soils to human management. Soil degradation is the decline in soil quality through physical, chemical, and biological processes that reduce productivity and ecosystem functions. It includes erosion, compaction, salinization, contamination, acidification, and loss of organic matter. Each form of degradation has distinct causes and management requirements, but all reduce the capacity of soils to support life. Erosion is the removal of soil by wind or water. Water erosion occurs when rainfall exceeds infiltration. creating runoff that carries soil particles. Sheet erosion removes small amounts uniformly across the surface. Rill erosion cuts channels that can grow into gullies. Wind erosion is most severe in dry, unvegetated, and finely textured soils. Erosion accelerates when protective vegetation is removed. Agricultural fields without cover crops are vulnerable between plantings. Cleared forests expose soil to rainfall impact. Overgrazed grasslands lose protective plant cover. Construction sites bare soil to wind and water. The rate of natural erosion and the rate of erosion under human use differ dramatically. The impacts of erosion include loss of fertile topsoil, accumulation of sediment in waterways, and reduced water quality. Eroded soil is the most fertile portion because it contains the highest organic matter and nutrients. Sediment fills ditches, reservoirs, and streams. Sediment-bound phosphorus and attached pesticides impair water quality. Compaction occurs when soil particles are pressed together, reducing pore space and increasing density. The most common causes are heavy equipment traffic and tillage when soils are wet. The compression destroys soil structure, reducing porosity and restricting root penetration. Wet soils are most susceptible because water cushions particles, allowing them to be pressed closer together. The impacts of compaction include reduced water infiltration, restricted root penetration, and poor aeration. Water runs off instead of infiltrating, increasing erosion and reducing water storage. Roots cannot penetrate compacted layers, limiting access to water and nutrients. Poor aeration restricts root respiration and microbial activity. Salinization is the accumulation of soluble salts in soil, primarily in arid to subhumid climates. It occurs when evapotranspiration exceeds precipitation, drawing water upward and leaving dissolved salts when the water evaporates. Primary salinization occurs from weathering. Secondary salinization results from irrigation with saline water and inadequate drainage. The impacts of salinization include osmotic stress, specific ion toxicity, and impaired structure. High salt concentrations make it difficult for plants to extract water. Specific ions like chloride and sodium can accumulate to toxic levels. Sodium disperses clay particles, destroying soil structure. Contamination occurs when substances not normally present in soil are introduced in harmful concentrations. Industrial activities release heavy metals and organic compounds. Agricultural activities can add pesticides and nitrates. Urban activities add oil, heavy metals, and salt. Contamination can persist for decades or centuries. Acidification is the decline in soil pH that occurs in humid climates. It results from the leaching of basic cations and from the addition of acidic compounds from decomposition and human activities. It is accelerated by nitrogen fertilizer use, which adds acidity. When nitrates leach, acidification mobilizes aluminum, which can be toxic to plants. The impacts of acidification include aluminum toxicity and reduced nutrient availability. Soluble aluminum damages root tips, impairing water and nutrient uptake. Phosphorus reacts with aluminum to form insoluble compounds unavailable to plants. Loss of organic matter occurs when decomposition exceeds inputs. Tillage accelerates decomposition by mixing oxygen into soil. Removing crop residues removes carbon inputs. Absence of living roots reduces rhizodeposition. The decline of organic matter reduces cation exchange capacity, water holding capacity, and aggregation. Reversing degradation requires addressing the causes and rebuilding soil. Erosion control uses cover and barriers. Compaction is relieved by tillage at proper moisture and by reducing equipment traffic. Salinization is managed by leaching salts and improving drainage. Contamination requires removal or remediation. Organic matter is rebuilt by adding organic inputs and reducing tillage. Prevention is easier than remediation. Managing to maintain soil cover, organic matter, and structure prevents most forms of degradation. Once degraded, soils may require decades to centuries to recover full productivity. The value of healthy soils justifies investment in management that maintains their quality. The rhizosphere is the narrow zone of soil surrounding plant roots, where root exudates and microbial communities create unique conditions. It is the most biologically active region in soil, containing higher microbial populations and more dynamic chemistry than bulk soil. Understanding rhizosphere processes is essential for improving nutrient use efficiency and plant health. Root exudates are compounds released by living roots into the surrounding soil. They include sugars, amino acids, organic acids, enzymes, and other compounds that feed soil microbes. The composition and quantity of exudates varies with plant species, plant age, and environmental conditions. Estimates suggest that 5 to 20% of photosynthetically fixed carbon is released through exudation. Organic acids and root exudates solubilize nutrients, particularly phosphorus and iron. Citrate, oxalate, and malate form complexes with aluminum, iron, and calcium, releasing bound nutrients. This mechanism is particularly important in nutrient-limited soils, where plants must compete with soil minerals for available nutrients. Exudates also modify the soil microbiome. The compounds released select for microbes that can use those specific compounds, shaping the community composition near roots. Some beneficial bacteria are attracted to root exudates, colonizing the rhizosphere and providing benefits in return. Others may be pathogens that respond to the same signals. Mycorrhizal fungi extend the rhizosphere dramatically. The fungal hyphae explore far beyond the root depletion zone, accessing nutrients in soil volumes that roots alone cannot reach. The plant supplies carbohydrates to the fungus, typically 20% of photosynthate, and receives phosphorus, nitrogen, water, and protection from pathogens in return. Rhizosphere bacteria produce compounds that affect plant growth. Some bacteria fix nitrogen, producing ammonia available for plant uptake. Others produce plant hormones that stimulate root growth. Still others produce antibiotics that suppress pathogens. The rhizosphere microbiome can be selected for beneficial functions through crop rotation and management. Root hairs increase the surface area for water and nutrient uptake and increase the volume of soil explored. The rhizosphere zone effectively extends as far as root hairs reach. Different plant species vary dramatically in root hair length and density, affecting the efficiency of nutrient uptake. Root colonization by beneficial microbes provides multiple benefits. Nitrogen-fixing bacteria reduce the need for fertilizer nitrogen. Mycorrhizal fungi reduce the need for phosphorus fertilizer. Disease-suppressive bacteria reduce the need for pesticides. These benefits can reduce production costs and environmental impacts. The rhizosphere is also the zone where pathogens concentrate. Root pathogens respond to the same exudate cues as beneficial microbes, locating roots by following exudate gradients. Some pathogens produce compounds that interfere with beneficial symbionts. The balance between beneficial and pathogenic rhizosphere communities affects plant health. Rhizosphere processes can be managed to improve plant health. Cover crops provide living roots that maintain rhizosphere activity through seasons when cash crops are absent. Crop rotation selects for different rhizosphere communities. Organic amendments support beneficial microbes. Reduced tillage preserves mycorrhizal networks. The concept of the rhizosphere extends beyond the root zone to include the hyphal networks of mycorrhizal fungi. These networks connect plants, allowing carbon and nutrient transfer between individuals. Mature trees may support seedlings through mycorrhizal connections, a form of below-ground cooperation. Understanding rhizosphere processes has practical applications for agriculture. Seed treatments with beneficial bacteria can establish beneficial rhizosphere communities. Foliar applications of some compounds can modify root exudation patterns. Breeding for root characteristics can improve nutrient efficiency. These applications reduce the need for external inputs. The rhizosphere is a dynamic interface between roots and soil, where chemistry and biology come together to determine nutrient cycling and plant nutrition. Managing this interface is key to sustainable agriculture that reduces external inputs while maintaining productivity. Soil and human health are connected through food, water, and exposure. The nutritional quality of food depends on soil nutrient availability. Drinking water filters through soil, acquiring minerals and being purified by soil processes. Direct contact with soil exposes humans to soil organisms that may be beneficial or harmful. These connections make soil health essential for human well-being. Food nutritional quality reflects soil fertility. Vegetables grown in fertile soils contain higher concentrations of vitamins and minerals than vegetables grown in depleted soils. The decline in nutritional quality of food over the past decades is linked to agricultural practices that deplete soil nutrients without replacement. Health recommendations to eat more vegetables assume adequate nutritional quality. Essential micronutrients, including zinc, iron, selenium, and iodine, come from soil. Plant foods absorb these elements from soil solution. Deficiencies are most common in regions with inherently poor soils or where nutrient depletion has outpaced replacement. Fortification programs address deficiencies that could be addressed through soil management. Soil contamination affects food safety. Heavy metals including lead, cadmium, and arsenic accumulate in soils contaminated by industry and persist for decades or centuries. Plants absorb these metals, particularly in acidic soils, and they enter the food chain. Maximum contamination levels in food are increasingly difficult to meet as soil contamination accumulates. Drinking water treatment relies on soil processes. Water moving through soil is filtered, purified by microbial processes, and minerals are added. Groundwater that is filtered through soil is typically cleaner than surface water. The purification capacity of soil is being challenged by increasing contamination loads. On-site wastewater treatment depends on soil processes. Septic systems rely on soil microbes to process waste, filtering pathogens and releasing clean water. The capacity of soil to process wastewater depends on soil type and system design. Failing systems can contaminate groundwater. Human exposure to soil occurs through dust inhalation, skin contact, and ingestion. Urban soils contain contaminants from decades of industrial activity. Children are particularly vulnerable because they ingest more soil through hand-to-mouth behavior. Soil exposures are linked to some chronic diseases. The soil microbiome contributes to human health through multiple pathways. Environmental mycobacteria that develop in healthy soils may provide immune training that reduces allergic and autoimmune diseases. The dramatic rise in immune disorders in developed countries may be linked to reduced soil exposure in childhood. Soil organisms produce antibiotics that have saved countless lives. Streptomycin, tetracycline, and many other antibiotics are derived from soil bacteria and fungi. Soil remains a source of new antibiotic compounds as drug-resistant pathogens evolve. The diversity of soil microbial communities may be essential for continued discovery. Composting and vermiculture convert waste into soil amendments through biological processes. These processes reduce waste volumes while producing valuable soil amendments. The finished products can be applied to soils, closing loops between waste and food production. Community composting programs build soil while reducing waste. Urban agriculture improves access to fresh produce in urban neighborhoods. Contaminated urban soils require management before food production. Raised beds with imported soil avoid contaminated native soil. Soil-based urban agriculture provides food, community building, and therapeutic benefits. Soil conservation addresses both human well-being and ecosystem health. Practices that build soil organic matter, minimize erosion, and maintain nutrient cycling improve both agricultural productivity and environmental quality. The investment in soil health pays dividends in food security and environmental resilience. Human health cannot be separated from soil health. The decline in soil quality that has accompanied industrial agriculture has contributed to declining food nutritional quality, increasing contamination, and possibly increasing immune disorders. Investing in soil health is investing in human health. As our story slowly comes to an end, Allow the final thoughts to fade gently into the quiet of the night. The rain and the calm ideas and the peaceful moments we've explored can settle softly in the background, like distant echoes of a quiet evening. There is no need to think about anything now. Your body can continue to relax, breathing slow and steady. If you are still awake, simply remain here a little longer and enjoy the calm atmosphere. And if sleep has already started to find you, let it arrive naturally and without effort. Thank you for spending this quiet moment here on Cozy Sleep Time. If you enjoy these calm stories and gentle explorations, you can return anytime for another peaceful journey into rest. For now, let the night grow quieter and quieter and quieter, and allow yourself to drift deeper into sleep.